Good morning, it's February 4th and this is your daily brief in science. Here's everything you need to know. A recent study has unveiled concerning levels of microplastic accumulation in human tissues, particularly in the brain, raising significant health concerns. Conducted by Matthew Campan's team at the University of New Mexico, the research analyzed tissue samples from 52 deceased individuals, comparing data from 2024 with samples collected in 2016. Notably, brain samples from dementia patients dated between 2019 and 2024 exhibited higher plastic content compared to samples from individuals without dementia collected from 1997 to 2013. Richard Thompson, a leading expert on microplastic pollution, highlighted the pervasive nature of microplastics in our environment, infiltrating food, water, and air, ultimately entering human tissues. The study suggests that microplastics primarily enter the human body through food, particularly meat, which may be contaminated due to agricultural practices. The most commonly detected plastic was polyethylene, found in 40 to 65% of liver and kidney samples, and 75% in brain samples, predominantly in nanoscale shards and flakes. Key unanswered questions remain regarding how microplastics enter the brain, their potential elimination from the body, and their health implications. This has prompted researchers to caution against complacency. While the FDA maintains that current evidence does not indicate a health risk from microplastics in food, the presence of these particles in the brain is particularly alarming. Professor Tamara Galloway noted a 50% increase in brain microplastics over the past 8 years, correlating with rising global plastic production. The average brain could contain approximately 4,800 micrograms of microplastics per gram, which is roughly equivalent to 0.48% by weight, similar to the weight of a plastic spoon. The findings underscore an urgent need for further research into the health impacts of microplastics, as their production continues to rise and is projected to significantly affect oil demand by 2040. Despite the study's significant findings, limitations such as small sample sizes and potential contamination highlight the need for cautious interpretation and further investigation. A new study from Durham University reveals alarming changes in the Greenland ice sheet, specifically an accelerated increase in the size and depth of crevasses due to climate change. This study highlights the critical relationship between glacier dynamics and climate factors, as crevasses form from uneven glacier movement, where varying ice flow speeds create tension that leads to cracks. While there was a temporary slowdown in the Surmet-Kujalleq Glacier, limiting crack formation to a 4.3% increase, the glacier's flow rate has now resumed, indicating the end of this equilibrium phase. Understanding the melting dynamics of Greenland's ice is crucial, as it significantly contributes to global sea level rise. Glaciology professor William Colgan emphasizes that Greenland has already contributed approximately 14 millimeters to global sea level rise since 1992. Projections suggest that meltwater could raise sea levels by up to 30 centimeters by 2100, with Associate Professor Tom Chadley predicting a potential sea level rise of 1 meter by 2100 and up to 10 meters by 2300. Concerns are growing that if temperatures remain above 2 degrees Celsius, Greenland and Antarctica could contribute to a sea level rise of 12 to 20 meters over millennia. In 2023 alone, Greenland's ice sheet lost 80 gigatons of water, continuing a trend of shrinking for 28 consecutive years. The study is the first to confirm the widespread expansion of crevasse zones across Greenland's outlet glaciers, with thousands of square kilometers becoming crevassed between 2016 and 2021. Lead author Dr. Tom Chudley expressed surprise at the rapid changes occurring in the ice sheet, contrasting with earlier studies that noted slower developments. The research utilized new automated methods for studying crevasses, addressing the dangers of in-person observation and challenges in satellite data analysis. Supported by the Arctic DEM project, this study emphasizes the importance of continuous monitoring using satellite data to understand the impacts of climate change on Greenland's glaciers. A recent study has revealed a surprising cognitive ability in bonobos, showing they can recognize when another individual lacks knowledge. This trait, once believed to be exclusive to humans, suggests that the understanding of different perspectives may have evolved much earlier than previously thought, possibly existing in our common ancestors to enhance cooperation. The research, conducted by a team from Johns Hopkins University, indicates that both humans and bonobos share a complex cognitive foundation, with these abilities likely evolving millions of years ago. The study published in the journal PNAS provides the first evidence that bonobos can assess another's knowledge level and communicate their own in collaborative situations. In the experiment, bonobos were presented with a scenario where a treat was hidden under one of 3 cups. While the bonobos could see the treat's location, their human partner was either informed or unaware. The bonobos displayed a significant awareness of the situation, indicating the treat's location approximately 8 times more often when a human was present than when alone. Interestingly, when the bonobos noticed that the human could see where the treat was hidden, they chose not to provide any hints. One bonobo named Kanzi was particularly driven by food, persistently trying to gain attention by tapping on the cage when the human could not see the treat. Co-author Chris Krupinier highlighted that recognizing gaps in knowledge is crucial for complex social behaviors, cooperation, and communication across species. Future research will further explore the motivations of apes and their understanding of the mental states of others, including whether they utilize gestures to influence their partner's beliefs. Chinese researchers have made a groundbreaking advancement in reproductive biology by successfully creating mice with 2 fathers. This achievement marks a historic moment in the field of genetic engineering, as detailed in a study published in the journal Cell Stem Cell. The research involved manipulating 20 key genes to enable male-only reproduction, overcoming previous genetic barriers associated with unisexual reproduction among mammals. The complex genetic engineering techniques employed included gene imprinting, deletion, and editing, which allowed researchers to bypass the usual requirement for genetic contributions from both a mother and father. By precisely modifying imprinting genes, they created embryos containing DNA from 2 male mice, which were then implanted into the uteruses of female mice. While the bipaternal mice were larger than typical mice, they faced significant health challenges, including reduced lifespans and sterility, with only 11.8% of viable embryos developing to birth. Despite these issues, the genetically modified mice displayed improved health and longer survival compared to previous experiments, indicating a major advancement in genetic engineering. Researchers plan to refine their techniques and explore applications in larger animals, such as monkeys, to enhance embryonic stem cell development outcomes. The ethical implications of this research for potential human applications remain uncertain, particularly concerning reproductive practices among gay couples and hereditary genome editing. If adapted for human use, this technology could redefine family structures and challenge traditional views on parenthood. It opens up possibilities for homosexual couples to have biological children, a concept that previously seemed unattainable. The findings could also lead to advancements in fertility treatments, genetic disease prevention, and regenerative medicine, although the prospect of human bipaternal reproduction remains speculative. This study raises critical questions about the future of unisexual reproduction in mammals and emphasizes the restrictions on applying such technologies to human reproduction. Researchers at Johns Hopkins Kimmel Cancer Center and the Bloomberg Kimmel Institute for Cancer Immunotherapy have unveiled a new computer model, named MANASCORE, designed to identify tumor-fighting immune cells in lung cancer patients receiving immune checkpoint inhibitors. This study, published on February 3, 2025, in Nature Communications, differentiates itself by utilizing only 3 genes to pinpoint immune cells targeted by therapies, a significant reduction compared to existing models that often require over 200 genes. The MANASCORE streamlines the identification process of tumor-active T cells, making it both less time-consuming and more cost-effective than previous methods. Findings from the research indicate that patients who respond positively to therapy exhibit a higher proportion of stem-like memory T cells, which play a critical role in generating effective anti-tumor responses. These stem-like T cells are characterized by their ability to promote self-renewal and maintain long-term persistence, which contributes to sustained immune responses. Ongoing collaborations with other laboratories aim to evaluate the MANA-score's applicability across various cancer types, utilizing a database of single-cell sequencing data to uncover distinct responder T-cell characteristics. The model not only identifies immune cells targeted by therapies, but also sheds light on the differences in patient responses to immunotherapy. Understanding the reasons behind the variability in patient responses is essential for developing better treatments for those who do not respond to current therapies. Immune checkpoint inhibitors, including PD-1 inhibitors, work by reactivating T cells to combat cancer, yet the differences in patient responses highlight the necessity for further investigation into the underlying factors. Zhen Zeng, PhD, the lead author of the study, intends to examine how the proximity of T cells with the 3-gene signature to other immune cells may influence clinical outcomes. Senior author Kelly Smith, PhD, stresses the importance of studying these identified T cells to enhance biomarkers and targets for combination immunotherapy. It is also worth noting that the study's authors have disclosed various conflicts of interest related to commercial partnerships and patents associated with their research. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.